Good evening. My name is Mark Barnico, and I'm the Executive Director of the University of Chicago, Francis and Rose Yuen campus in Hong Kong, our premier location in Asia, representing UChicago values of free and open discourse, rigorous debate, and the exchange of ideas. Welcome to this series, 1.5 Degrees. We're broadcasting tonight's event live via Zoom, Facebook, and YouTube from the UChicago Yuen campus in Hong Kong. If audience members have questions to submit, you can do that through the questions and answer button by first registering on Zoom. I also encourage you to visit our website at www.uchicago.hk for the latest news and information, or follow us on the UChicago Yuen campus Facebook page. Tonight we continue 1.5 Degrees, which addresses the significant threat of climate change and what we can all do about it. This week, we begin our mini-series program with the University of Chicago's Marine Biological Laboratory Director, Dr. Nipam Patel, who will share the 133-year history of the MBL. Let me first begin by introducing Dr. Patel. Nipam Patel is Director of the Marine Biological Laboratory and a professor at the University of Chicago. He joined the MBL in 2018 from the University of California, Berkeley, where he was professor and co-chair of the Department of Molecular and Cell Biology and professor in the Department of Integrative Biology. Dr. Patel is the 20th scientist to serve as MBL director since its founding in 1888. Dr. Patel grew up in El Paso, Texas, and received an AB in biology from Princeton University and a PhD in biology from Stanford University. Prior to joining UC Berkeley in 2003, he was a professor of organismal biology and anatomy at the University of Chicago, a Howard Hughes Medical Institute investigator, and a staff associate in the Department of Embryology at the Carnegie Institute in Baltimore, Maryland. A longtime member of the MBL community, Dr. Patel has taught in the MBL Embryology course since 2001, and served as course co-director from 2007 to 2011. He's a leading scholar in modern evolutionary and developmental biology with specific focus on the evolution of body patterning and segmentation regeneration of the germline and structural coloration. His scientific expertise encompasses the development of novel genetic model organisms for biological study, which can reveal much about human biology, and the application of advanced imaging technologies to probe the fundamental dynamics of living systems. Dr. Patel is an elected fellow of the American Association for the Advancement of Science and has been awarded numerous honors, including the Schubert Endowed Chair and the William V. Power Endowed Chair at UC Berkeley, the McKnight Scholars Neuroscience Fellowship Award, and NSF Predoctoral Fellowship. Other past appointments include faculty curator at the Essig Museum in Entomology, UC Berkeley, an adjunct professor at the National Institute of Genetics at Shizuoka, Japan. He's also the author of 130 scientific publications. Dr. Patel was an editor of the journal Development and serves on the editorial board of several other journals in the biological sciences. He served on numerous advisory boards, including the board of directors of the Society for Development Biology. Welcome, Dr. Patel. We're excited to learn more about the history of the MBL this evening. I'll turn the floor over to you and then rejoin to facilitate a conversation and take questions.
Thank you. It's a real pleasure to be here. I thank Mark, and I thank the rest of the UN campus for this invitation. And it's a real pleasure to be able to introduce everyone that may not be familiar with the Marine Biological Laboratory. So we are now part of the University of Chicago and are a premier, not just a marine biology lab, but really a research site for basic science. And so I hope to share with you today a little bit about the history of the MBL, what makes us special, and then to introduce you in a bit to how we actually participate in marine biology and the ways in which we are involved in researching climate change, though that will really follow in some subsequent lectures in this series. So I'm going to go ahead and start my screen share here. hopefully that all looks okay. So again, it's a pleasure to be here. And as Mark said, I am the director of the Marine Biological Laboratory. So first of all, where are we located? So, many of you know that the University of Chicago is right here in Illinois on Lake Michigan. But we, the Marine Lab of the University of Chicago now, are located all the way here in Cape Cod, Massachusetts. So actually a little ways away from the University of Chicago, but in a great location on the ocean. So this is a view from space of where we're located. So this is this little boot that goes out from the state of Massachusetts. And the MBL is located right here, kind of the heel of that shoe. And this is an aerial view of our campus. So we have a pretty substantial campus here right on the water. And we have a number of buildings that are really dedicated to our two primary missions, which are research and education. And it's an ideal location for what we do. So a little bit about our founding. So our history really dates back to about 1871. So that's old by certainly American academic standards. And it began with the establishment of a United States Department of Commerce Bureau of Fisheries here, right basically across the street from us. And then in 1885, Spencer Fullerton established a permanent research station for the Fish Commission in Woods Hole, Massachusetts, the village that we are in. And then the important event really was 1887, where the Women's Education Association in Boston appointed a committee of 12 members to set out plans for organizing a permanent seaside laboratory that would combine research and education. And we became incorporated in 1888. So our first laboratory head was an embryologist named Charles Otis Whitman. And at that time, he was at Clark University, but he was soon to join the University of Chicago, and he was our the first director of the MBL. So the MBL actually has a very old connection to the University of Chicago. In fact, the first three directors of the Marine Biological Laboratory were faculty at the University of Chicago. But why are we located in this particular location? So why did the fisheries want to set up a site here? And then why eventually was it decided that this was a great place to set up a marine lab? And it really has to do with the environment, the ocean environment in this area. So in this part of Cape Cod, there's really a confluence of ocean currents. So there's cold water coming down from the north and the Gulf Stream coming up from the south. And these currents mix in this area right around here. So what that means is that this is a very nutrient-rich area of the ocean. So if you were to go diving here, you would not be impressed. So the water is pretty murky looking, and you wouldn't. It's not like your picture of a beautiful coral reef that you might imagine. Instead, it's, like I said, a very nutrient-rich place. And what that means is that there's an incredible amount of biodiversity here. So if your goal is really to study the ocean and to study life in the ocean, then this is really an ideal place to do that. So this slide just highlights some of the diversity of organisms that you can find in a place like Woods Hole. So everything from really large mammals. So, we have populations of whales that come here to feed every year, and you can take a boat out and see that amazing thing, all the way through, fishes that come through here, but then all sorts of smaller organisms that probably are a little less known to you, including things like the squid that we have here. There's actually a species of coral that manages to survive in the water around here, skates and horseshoe crabs, all sorts of creatures, and then an incredible diversity of really microfauna as well, very small invertebrates. So since our founding in 1888, we've really served as an institution, as I said, for both research and education. So the courses really began right at the beginning. And so one of the things we really are proud of is our long-standing commitment to educating people in students and the public in marine science and in basic biology. So those classes actually started right at the beginning. So there were 17 students and investigators in the inaugural year, but that grew to 110 in only 4 summers. And it was primarily at that time in the summers that we ran our classes, and that's still true to this day, but I'll tell you a little bit about the ways we're expanding that. One of the things to note is that there was a very good representation of women here from the start, and I think that has a lot to do with it being founded by that women's board in Boston. So the first summer courses were in things like invertebrate zoology, which has now become a course called neural systems and behavior. And it very quickly were added to by other courses such as embryology, physiology, and courses in microscopy techniques. And many of these courses, particularly embryology and physiology, are still going to this day. So these courses are now over 130 years old. So there were also a core group of investigators that would come here in the summertime to do all sorts of work. And a lot of areas were pioneered here. So like the interplay of evolution and development. Thomas Hunt Morgan, which many of you might be familiar with, he was a faculty member at that time at Columbia University, but he would come here for the summers, and he came for about 50 years in the summertime. And he would bring his fruit fly lab here, and Drosophila melanogaster is what he worked on. And eventually, he went on to win the Nobel Prize in 1933 for his work in Drosophila, some of which was done here at the MBL in the summertime, but he also came in the summer here to explore lots of other areas of biology that fascinated him. So again, this very long history going back in time of MBL's dedication to both education and research. So we became affiliated with the University of Chicago in 2013. And this is a quote here from Bob Zimmer, about the MBL. The MBL has an extraordinary valuable role as a source of innovation and creative collaboration in modern biology. And I think that's really the heart of what we do. So this is a place where researchers come from all over the world and bring all of their creative energy every year and collaborate with one another to make incredible discoveries. So in all, about 59 Nobel Prize winners have been affiliated with the MBL. And when I mean affiliated, we don't have that size faculty here. What we have, though, is programs that bring them here as educators and as collaborators in scientific discovery. And many of them will, in their Nobel Prize acknowledgements, will highlight how important MBL was in their career. Some of the key discoveries that really MBL has played an important role in include understanding the mechanisms that explain nerve impulses. So it was working with giant squid axons here at the MBL that people began to understand how it was that neurons actually send impulses down nerves. And so that pioneering work on what are called ion channels and how that works was done here at the MBL. In medical research, a very important reagent is found from horseshoe crab blood. So it's how you test whether or not there's bacterial contamination. It's still used to this day to look for bacterial contamination in medical devices and vaccines. And that was discovered here at the Marine Biological Laboratory. Proteins that control cell division, including ones that are now known to play important roles in cancer, those were discovered here at the MBL in the summertime courses. And how molecules are transported within cells is another area of research that was pioneered here at the Marine Biological Laboratory. In 1902, there was almost a major change in the direction of the MBL. So it considered becoming a research arm of the Carnegie Institution of Washington. But in fact, Whitman, who was director still at the time, rallied against this. And he really argued that it was really both education and research that were critical, and becoming part of the Carnegie Institution would make it a research-only institution. But I firmly believe that that combination of research and education has really been key to the MBL's success. And as Mark said, I've been coming here to the MBL. I think this is my 22nd summer now. I've only been director for 3 years, but I've been coming for a long time. And that primarily has been through that education mission. So I mentioned, for example, some of those early courses. The one I participated in quite a lot is the embryology course, and it started in 1893. So here you can see that class from way back then, and it's still going to this day. Here's a slightly more modern picture. You can get an idea of kind of the importance of this course in the field. So for example, in this class picture from 1979, you see Tim Hunt, who won the Nobel Prize in 2001. And so Tim was one of those people that really pioneered the work on understanding proteins that are involved in cell division. And another one of the students in the class was Joan Ruderman, who went on to become the first female MBL director. Well, this is a little bit more recent picture of the MBL embryology class. The key to our courses is really the way we run them. They're not just didactic courses where students are following a recipe book. So in these courses, we have amazing equipment available to them, amazing reagents, and faculty from all over the world that come to teach them. In fact, in something like the embryology course, in a typical summer, there are 24 students, and about 50 faculty to teach them. So an amazing student to faculty ratio. And the idea is we teach them how to do basic things at the start of, we divide the courses up into little modules. And then the students are really encouraged to go out on their own and do their own projects with our help. And it's fantastic that students are what they're able to do. One of the things we really encourage the students to do, and this may seem kind of a strange thing, is we encourage them to fail. So we tell them if they're not trying and failing, they're not really trying hard enough. And so we get them to do incredibly difficult experiments. Often they don't realize how difficult they are, and they get them to work. And it's a fantastic experience. So embryology, of course, is not the only course we teach here. So this is a list of the courses that we offer. So in a typical summer, there's usually 21 to 23 of these advanced training courses. Now, these courses are primarily directed towards PhD students and postdocs that are in labs. And they range in this, large diversity of areas of science, from things like developmental biology, embryology, to parasitism, microbial diversity, physiology, lots of courses in the neurosciences. And another thing that we specialize in is imaging and microscopy, and we have a long tradition of both excellent research and education in microscopy. So these courses have become world-renowned, and in many areas of biology are the course for students in that area to take. And again, when I say students, I mean generally PhD students and postdocs, occasionally young faculty as well. One of the things we're trying to do now at the MBL in the last few years is to take that ability that we have to educate and expand that. So to have new education opportunities at the MBL. And the University of Chicago has been really instrumental in helping us expand in this direction. So one of the things we've done is actually open up our courses in the non-summer months to high school, secondary school students. And it's an amazing experience to be able to have them use all of this fancy equipment and to teach them what research is really about. So they learn about, science in their textbooks, but that's just usually a lot of facts. And the thing we're really interested in, excited to teach them, is how scientists do research and the kinds of things that are cutting edge. And we really give them an opportunity to do quite amazing things. So I've been involved, for example, in teaching imaging microscopy to high school students. And this just illustrates, this is a fruit fly embryo, but it illustrates the kind of imaging they can do. And then we have them do 3D imaging reconstructions. And this was actually done by a high school student in one of our labs. And you can see this is showing the nervous system and the support cells in the nervous system. Again, it's in a fruit fly, but it's an easy system to teach them, and they learn this sort of imaging that's really at the cutting edge of biomedical research. So it gives them a chance to do that. In addition, we have a long-standing, wonderful course called the Semester in Environmental Sciences, where we teach them all about ocean research and marsh research and climate change and things like that. We've been expanding that to also now include undergraduates from the University of Chicago. So again, we were fortunate to be able to actually run in-person courses here for University of Chicago students, both in the fall of 2020 and in the spring of 2021. So we just sent some of those students home just a couple of weeks ago. And again, we gave them opportunities to really be able to do cutting edge research as part of their education. So in learning how to do these kinds of things. And then, we also have a very outstanding faculty at the MBL. We have about 25 resident faculty. We hope to expand that to about 30 in the next few years. And these are some of the areas that they work on. So you'll hear quite a bit later in this series about the work that goes on in our ecosystem center that really focuses on ecology and climate change and coastal environments and ocean environments. But in addition to that, we have a center here that's really focused on microbial organisms, so microbes and their importance to the ocean, the planet, and human health. And then we've specialized for a long time in biological discovery and marine organisms. So we really do take advantage of the fact that we are on the ocean. So in addition to just doing basic science, we do that, but we often do it with marine organisms. And doing it with marine organisms is fantastic for a couple of reasons that I'll go into. One of them is that diversity of organisms here, again, is fantastic. And so we can really take advantage of that to do incredible exploration in biological systems. And finally, as I mentioned before, we have real expertise in cutting edge imaging and microscopy. And so that's something else that our researchers are involved in. We have two big initiatives that are going on right now in our research area. One is development of new genetically tractable organisms. So many of you may be familiar that a lot of biological discovery goes on with a relatively small number of organisms that actually have proven to be very extremely useful for basic biology and modeling human disease as well. But there's a clear need to really expand that set of organisms. This current set of organisms doesn't represent that much of biological diversity, and there's a lot of really interesting biology that's better studied in other organisms, particularly marine organisms. So we're really working on solving problems of husbandry of those organisms and making them genetically tractable. And what I mean by genetically tractable is being able to do things like edit their genome and to be able to do manipulative experiments of that kind. And then also to expand our imaging initiative. So MBL has a lot of breakthroughs in its history in microscopy, and we're trying to take the lead on continuing that. So as I mentioned, in imaging, we've been able to do amazing things in our history. For example, Shinya Inoue was one of the first people to be able to really watch cells dividing in real time and to see how chromosomes segregate. So we're taking on many of the challenges and trying to improve imaging. So this pyramid just sort of represents the pulling forces on trying to do cutting edge imaging. That, you know, for in order to see things better, you use more light, but that tends to kill things. And so you want to have a balance of that. And then the longer you image, the harder it is to do. And so there's a lot of competing forces when you're trying to do imaging, and we're trying to sort of expand this pyramid by improving all of these things to be able to build better and better microscopes that can make new and exciting discoveries in biology. And then in the area of new research organisms, as I explained, we're trying to really expand the group of organisms that scientists can use in biological discovery. And a really good example of how expanding this is very important to biology comes from the field of regeneration. So many of the organisms that scientists are used to using for biological systems, like mice, for example, they're great systems for a lot of areas of biology, but one thing they don't do very well is regenerate. And so it turns out that there are a lot of marine organisms that regenerate incredibly well. So like sea stars, you can cut the arm off, they grow their arm back. Same with something like an octopus. There are even organisms that can regenerate their guts and then regenerate all sorts of other tissues that are marine animals. So we think that it's important to expand the repertoire of organisms that biomedical research uses to include marine organisms like that we think can hold the key to many important discoveries. And in fact, history has taught us that many of those important discoveries will come from these kinds of marine organisms. Like previously, I highlighted the importance of squid in understanding neural conduction, and that's not just specific to squid, that's how we work as well. So just very quickly, just a couple of those kinds of organisms we work on. My own lab, one of the animals we work on is this little crustacean. And one of the areas we're really interested in is how they get the right kind of limbs on the right segment. And we're able to actually genetically manipulate that to change that pattern. But this isn't just an esoteric exercise in how these animals develop. These same genes are involved in setting up your body plan as well. And so by doing these experiments in these crustaceans, we actually understand for all organisms, for all animals, how this process works. Here's another animal that we work on here now at the MBL called Nematostella. It's a sea anemone. Again, it's a real champion at regeneration. You can cut it in little pieces and each piece will regenerate. So we're very interested in understanding the biology. And in my last slide, just show you that one of the animals that we've really spent quite a bit of time in the last few years developing are cephalopods, are squid, octopus, and cuttlefish. And this just shows the one that we've really focused on. It's a little animal called the Hawaiian bobtail, or sorry, a rainbow bobtail squid. And Euprimna berrii, and it's actually a pretty small little squid and easy to raise. And so we've been doing the husbandry, but we've been able to genetically manipulate them. And so this is an animal in which we've removed one of the genes that's responsible for making the pigment in it. So it's an albino now. And so we really hope to use these animals to pioneer both neuroscience research. So there's amazing questions about how they've evolved complex brains like ourselves, but in a very different way. And then understanding how they control their tentacles. They actually have a decentralized brain that goes down their tentacles. And then understanding things such as how they're able to regenerate their arms. And so those are just some of the areas of biology that we study. And as I said, you'll hear from other people at the MBL a lot more about the specifics of the climate work that we do as well. So with that, I will end, and thank you all very much.
Thank you, Nipam. That was great. Great overview, great introduction. I'm going to start with a question that just came to my mind as you were presenting your slides, which is around the globe, how varied are these organisms that you're dealing with? Are you finding some of those same organisms on the Atlantic coast as you would find in on the Pacific coast and throughout Southeast Asia?
Yeah, absolutely. So, I mean, one of the interesting things, of course, that you have with the ocean environment is that in some ways it's very easy for organisms to travel around the globe, but you also do get ones that are very localized to particular areas. And so, one of the things, in fact, that we're very excited to do is to collaborate with marine labs all over the world, because we think that's also key to developing this sort of research community. And so we've had very strong ties, for example, in parts of Asia, and Asia has really led the way with some beautiful pioneer marine labs. We have close ties, for example, with the Okinawa Institute there in Japan. And I'm really excited to build close ties with some of the marine labs in Hong Kong, especially given some of the incredible technology that they possess that could be really instrumental in advancing our research.
Great. One of the questions that just came in is there an MBL equivalent in Hong Kong? Can you say who that is, or the ones you just referred to?
Yeah, there are a couple labs in Hong Kong that have marine facilities. I'm sorry, I'm suddenly blanking on some names here, but we came to visit there about a little less than 2 years, I guess about 2 years ago. And we're really excited to actually work with a whole consortium of the labs there in Hong Kong, who, because of the location of Hong Kong on the water and the importance that the ocean plays, not just the Hong Kong, but of course all of Asia being very tied to the ocean environment. We're really excited, and there's a lot of interesting work going on with preserving the coastal environment, both as humans impact it and as sea level rises. And I think you'll hear quite a bit more about that as this series goes on.
You mentioned that there was some maybe specific technology, unique technology here in Hong Kong. And it was one of the questions actually as you were going through your presentation that struck me that especially with all this advanced imaging that's going on, do you have close connections with corporations that are developing new technologies as well? How does that work?
Yeah. So we historically have had very good ties with quite a number of microscopy companies. They help to provide us with commercial equipment for our education, as well as we help to provide them information that on the systems that we're developing. And so, in particular, we have very close ties with Olympus, with Leica, Nikon, and Zeiss over the years, and they're all here on campus right now, helping to provide us with some of our imaging technologies. And we're real interested in our imaging initiative really coupling with these corporations, because ultimately anything we develop, it's really up to companies like that to actually then commercialize and to make available to everybody else.
This particular question may be covered in the next couple episodes, but I'm just curious, with all the organisms that MBL is dealing with, is climate change having an impact on those organisms at that level?
Yeah, it definitely is. It changes what's available at different times of the year, and we've really seen that over time. So, animals that would normally have not been here till, say, July and August because of the water temperature are now showing up a month early. So you're definitely seeing the impacts. And we are very concerned about the impacts of sea level rise as well, because we basically are at sea level. And so that's something that we are, working on to understand how to harden our infrastructure, and working as a community, because we can't solve that problem just as our little, institution here. So we're working with the entire coastal community here in the state of Massachusetts to really look hard at how we can protect our labs and the people that live here and everything.
Thank you. There's one question here from one of the audience members. It may be specific, and I'm not sure if you can answer it, but do you have any omics-driven research in marine microbiome?
Yeah, absolutely. So one of the centers we have here is the Bay Paul Center, and the Bay Paul Center has really pioneered being able to analyze genome sequences for microbiomes. So that's one of the cutting edge research areas that we're involved in. And we teach courses in that. So we have a course where people come in, they sequence the microbiome, and then they use our tools that we've developed to analyze what the makeup of microbes is, how it changes over time. And of course, we initially applied that to marine systems, but it's become increasingly important in really looking at human health and the gut microbiome. So yeah, we have the facilities for doing that kind of sequencing and the computational facilities for analyzing that kind of data. And so it's one of the areas that we're very proud to have taken a lead in.
Excellent. One of our audience members said the Swire Institute, part of HKU here, is probably who we you met with.
Sorry about that. I suddenly blanked on the name.
Yeah, no worries. I'm glad our audience member was here to remind us both.
Yeah,
actually.
no, and we had a wonderful visit, and I think it's it would be fantastic to increase the collaborations we have throughout marine labs like that.
Now, you talked about the high school students that were there. Can you elaborate a little bit more on the levels of students and the quantity of students that you're able to educate? And then how many are coming from Asia today? Like, how many people are you getting to apply for admissions to your programs today?
Right. Well, we've just started the high school program, so it's pretty new. And of course, it was just getting going when, of course, it got shut down by the pandemic. And so we're just getting it up and running again. So the way it works is that we can take high schools from anywhere in the world. And in fact, we did have a high school that was coming from China, and then the pandemic hit and they weren't able to travel. When we started, it was obviously, we started with schools that were in the US, but actually throughout the US. So we had schools coming from California and Texas. And so anyone can sign up to do it. It needs to be done. Students can't one-off sign up. So they have to have like a group of 10 or more students from their high school who want to do it. And it's an in-residence program. So we bring the students, we keep them here for a week in our dorms and feed them and give them a really intensive, immersive experience. So we give them some lectures, but really the highlight is working in the labs, getting to use our equipment, getting to learn about what scientists really do, and being able to do sort of cutting edge things. So we're excited to get back to that. And you can go to the MBL website and look up under education, and there's a contact information for the high school program. And we're very eager to have people from all over the world come, because we think it's, science research really only will advance significantly by being a worldwide community effort.
Excellent. One of the audience members actually just sent us their email. They're interested in the program for high school students, so I'm glad you addressed that. One of our, we're all about free and open discourse, right? So one of our audience members said embryology in med school was really boring and intensive. And how do you make it interesting at MBL?
Well, I think part of it, of course, is that probably in med school, embryology is largely, again, taught from a textbook, and it's, memorizing a lot of facts and names. And that's not, what I think makes embryology exciting. I have to say, I got very excited. I actually was able to take a course in embryology as a high school student. And the most fascinating thing is that we got to work with chick embryos. When I was, that was the easy thing to get. You go to the farm, you can get eggs. And I think watching living embryos is an amazing thing, right? So that's really the key, is to show students how miraculous biology really is, to watch living organisms, to try to understand interesting questions. So, I think one of the things we do is, like, you have like a frog embryo, you watch the cells divide, you suddenly see a heart form and start to beat. And there are an infinite number of questions you can start to answer or ask. But then we show students how to ask those questions and answer those questions. The modern day techniques of doing gene editing, of doing advanced imaging, of, all those kinds of manipulations. I think that's how we make something like embryology really exciting.
Great. This must be from a UChicago student. They're interested in taking classes at MBL, but they wondered whether they could do that during the regular school year of their programs for UChicago students. Yeah,
absolutely. So as I mentioned, we actually had students in the fall and the spring, and we intend on offering courses every spring now for University of Chicago students. And so they can contact the Biological Sciences Division and they can see the courses that we offer. So I think our next year, our plan is to offer a series of courses. We would actually have a physics class, an art class, and then a stem cells and regeneration course, an imaging course, and an oceanography course, as well as a new set of courses in neuroscience on synapse formation and neural structure. So we're going to have quite a number of courses starting next year, and we hope to keep expanding that.
Oh, that's fantastic. Taking the 30,000-foot view, how has this discipline grown, in the last 50 years? Have you seen a larger number of people in each cohort studying this particular area, and what's driving that interest?
Yeah. Well, I mean, certainly in the area of, studying ecosystems and climate change, I think people understanding the consequences of not dealing with it is becoming more and more obvious, right? And so a lot of young people are very interested in being able to help make a change in what's going on in the world. And so they're becoming quite interested in being trained in marine biology, coastal ecology, and things like that. So we've seen a real interest in that as people understand how important it is to understand human impact and to do something about it. But a lot of the other things we do in basic biology, they continue to be quite fascinating. We change with the times as well. So as areas of biology evolve, our courses evolve. So we start to do different things. So for example, 30 years ago, you wouldn't really have a stem cells course and a regeneration course. We didn't know much about those things. But now as we know more about it and can apply modern methods, those are new courses that will pop up. And so we have a new crop of people who are really interested in that particular area.
That's great. I mean, that ties into one of the audience questions. We have some really informed people here. So great questions coming in. Are you by any chance using CRISPR-9 on genetic manipulation? Do you highly recommend CRISPR-9 to genetic manipulation? And then there was another question that related to ethics. And I thought maybe that would be good to kind of merge those two questions.
Yeah, absolutely. So in fact, one of the, one of the high school courses we're teaching now is actually on CRISPR-Cas9 genome editing and understanding that. So I think the public is really interested to know a lot more about it because you hear about it in the news all the time. So I can answer the question in a couple of ways. So as a basic science tool, it's fantastic. So it allows us to take organisms that we would not have normally been able to study and to very easily be able to do manipulations we didn't think were possible, even 6 or 7 years ago. So I showed one example there, which was the little crustacean we work on, Parhyla. So it used to be you would find an interesting gene, you could look at how it's expressed and you could hypothesize what it might do. But actually testing that was very difficult. Now we can tell you in 10 days what that gene does. We can go in CRISPR-Cas9, we can edit that gene and tell you what it does. So we have a great time now showing students and letting them do it with their own hands. So in that case, we do it in frogs and zebrafish where development's very fast and they can see the results in just 1 or 2 days. And they can see the results of, say, knocking out a gene involved in eye development, affecting the eye. But there is a larger ethical question about, what should we be able to do with that kind of technology? And that really gets to the question of, should we be able to release organisms like that? And should we be able to do this on humans? And those are really, really important questions that really require a great conversation, not just by scientists, but by ethicists and the public. And so when we, teach these kind of techniques, again, we're just doing it on, a zebrafish or a frog or something. We're not releasing those organisms, but we discuss with people what it would mean to do those kinds of experiments and how people should come to those decisions. So I think it is really important to have very deep ethical discussions about those ethics, and we are very involved in that. Though our own particular research, we do not, make genetically modified animals which we're intending to release, and of course we don't do direct experiments on humans, but we are involved in those conversations, and I think they're important conversations to have, and there are pros and cons to all of that.
Do you have, going back to the question about corporate relationships, do you have downstream relationships with pharmaceutical companies that are benefiting from your research?
Yeah, a little bit. I mean, so we've had, like, for example, companies that would like to use our advanced imaging capabilities to do drug screens. for example. So, you can say, oh, well, I could do this drug screen in mice, but that would take me, 10 years, and, all of these mice. But people are coming up with inventive ways to be able to do it on cells in culture and to target specific, say, biochemical pathways and just screen them by imaging. So that's sort of an example where we do that. There's always, obviously a lot of interest in farming, essentially farming for new compounds in marine organisms. So as we expand our reach of new research organisms, I'm sure there'll be a lot of interest in asking whether those organisms produce any compounds that could be beneficial for human health.
There seems to be a lot of interest here from Hong Kong. Another attendee mentioned that the Hong Kong University of Science and Technology, HKUST, also has a good marine biolab located here, located in China, actually. I'm not sure the location. Maybe they'll put that in the Q&A box or the chat box. One of the questions was around overfishing, and with the work that you're doing, are you seeing the impact of overfishing on the oceans, and is that affecting your work?
Yeah, so that's not so much of what we do. So we're next door to the NOAA, National Oceanic Atmospheric Association and Administration, sorry. And so they're responsible, for example, for keeping track of fisheries and things like that. But interestingly enough, you say that we have a new project that we're doing. So there's a pond right next to us, right? We're on the water, and then there's an open pond to the ocean, and that's where our boat comes in. And when we do our squid collection, excess squid get thrown back in the water, and there are these striper fish that come in to eat them, and they know that we're going to do that. And they're important in the fishing industry, and they're really something that anglers around here are really excited about. And so we've actually started a little project tagging them and trying to understand, their migration and their patterns and things like that, in order to really inform fisheries science about how to improve controlling that fishery and things like that. So it's something that we don't traditionally do, but we see it as an interesting thing that we could bring some of the science we're good at to bear on. And it's also a great way to engage the public to think about, how complex the ocean is and the ways that we need to manage it, and to really understand the life of the organisms out there. So I think, you see with fisheries, it's not just like how you're catching the adult fish and how many you're catching is, do you know where they go to breed? And are you preserving those locations as well? Because if the adults have nowhere to produce young, your fishery will collapse, even though you're not fishing the adults.
Mm-hmm. That may be a good lead into the next question, which is, does MBL research coral reefs at all?
A little, I mean, not. We, of course, are not on a coral reef, but we have researchers that will go to some of those areas and do work on tropical reefs as well. So we are doing some stuff with coral. We are using one of the native corals that's here, but of course, we also are very interested in using that information to help with coral reefs in more tropical areas. And then we have in our cephalopod research, some of our cephalopod researchers actually spend time in reefs in Indonesia, for example, looking at the behavior of octopus in those areas. And so we do have researchers that spend quite a bit of time out in more tropical coral reefs.
Great. One of the questions from the audience is, what are the biggest hurdles to getting scientists to use new marine organisms as models in their research? Is that an issue?
It is. So, you can get people a little bit, you can show them some really cool marine organisms. They get a little bit excited, but there are some significant hurdles. So one of them is the problem of husbandry, of taking care of the marine organisms. So anyone that's tried to keep a reef tank, it's a lot of work. So a lot of work goes into keeping the tank environment good. And so one of the things that we're doing, and this is something that we're really just getting ramped up, is to develop automated systems to take care of those animals. So often when you start working on a new animal in say a relatively small lab with not a very big community, you spend a lot of money just hiring people to keep the animal alive. And so we're trying to automate those systems and to improve the rate at which they reproduce so that it's easier for people to keep that animal. And then to do some of the preliminary legwork of getting gene editing, for example, to work in that organism. So in fact, we now have some funding to really develop that for quite a number of new research organisms in the hopes that then we can get more people to adopt them.
I see. One of the audience questions is, what is the most advanced microscopy technology available
Oh,
to
it,
MBL?
yeah, it's going in a lot of different directions at one time. So one of the areas that we're specializing in is light microscopy. And one of the innovations over the last few years is a technique called light sheet microscopy, which is actually a very old technique that was described a long time ago, but has now become a very powerful tool. So it's to use sheets of light moving very rapidly to do very rapid, very fast imaging that's very gentle for the organism. So now you can image entire organisms in just a second. So you can image over and over again. It's very important if you're looking at, say, imaging how neurons are firing in a whole animal as it's behaving. And so up until a few years ago, it was very difficult to do something like that. But here at the MBL, we're developing several of these light sheet microscopes that can hold a variety of sizes of organisms to do that kind of thing. There's also the area of super resolution microscopy. So forever, we were all taught that there's a resolution limit to light microscopy, that once you get down to the wavelength of light, obviously you don't have better resolution than that. But physicists have come up with amazing tricks to go way beyond that level of resolution. And so there's some fascinating things where people can now track single molecules in a living cell as it's developing, as it's alive. And so those just open up completely new avenues. And so those are some of the things that we're really excited about to be pushing on the cutting edge of the MBL.
I think you're whetting the appetite of so many of the people on this program. I'm delighted to see that. We had one question, are there postdoctoral opportunities for foreign students at MBL? And maybe what's the path for applying for those kinds of programs? And another one was, how can we join with your lab? And maybe that kind of leads to a broader question of, how do students apply? And then who should they contact? How should we put them in touch with you?
Okay, I'll start with the postdoc one. So, MBL scientists are always looking for talented postdocs to come join the lab. And so we often put out ads. So if you go to the MBL website, you can see research opportunities and employment opportunities. And so when labs have open positions, they post them. But I think labs are often also very open to getting solicitations from people who know about the work that you do in your lab and have a particular interest in that work. And so we're always open to hearing about that. For other education opportunities for advanced training courses, again, at our education website, you can see the application process. So each course has its own application portal. You put in your materials and we accept students every year to these courses. I will say admission is very tough. We get, far more applicants than we can take. So sometimes people have to apply a couple years in a row to get in, but it's well worth it. I think that those are
are there certain credentials or qualifications or thresholds that student applicants need to meet generally?
Yeah, I mean, it varies quite a bit, but I think one of the things is that we really want to find students that are going to benefit greatly from the experience, right? And and they can be at all sorts of levels. So we often have like first-year graduate students taking an advanced course because they're just starting their career and they want to know like what are the techniques that would be available to them in their research area. Other times it's students that are trying to make the transition from being a graduate student for a postdoc, and they're already very well trained in one system, but they want to learn about other things. In terms of credentials, I mean, it's really a lot of it depends on the letters of recommendation that you get and those kinds of things.
Got it. Somebody is asking you if you'd be willing to speak at another university in Asia at some point in time. So
yeah, absolutely.
Is that something you're willing to do?
Yeah, you can get in touch with me, and as we begin to either by Zoom or travel in person again, I'm happy to entertain that. I make a couple international trips a year, so I can weave it into another trip.
Great. And then one of the audience members is asking how MBL is funded and whether it's through endowments at UChicago and support, or and just in terms of the overall funding of the mission of MBL. How does that funding work?
Yeah, so in like any other institution, it's a lot of different things. So we do have our endowment, and that helps in some ways to fund us. We have significant philanthropy from a really wonderful group of individuals all over the world that support us. But then we also depend a lot on federal grants. So we have grants from the National Institutes of Health, the National Science Foundation, Department of Energy that fund our researchers here. Our courses are funded in large part by the Howard Hughes Medical Institute and the Burroughs Wellcome Fund, and then in addition to federal grants that actually provide significant support for all of our courses.
Okay. The same person that was thinking of inviting you to speak at their university in Asia is also asking whether there are any research grants that fund collaboration based with other universities in Asia.
Yeah, there aren't specific ones at the moment, but we are really excited about doing that. We have had some programs in the past with specific universities to do exchanges, and so we'd be very excited to set things up like that again. And I know that with the University of Chicago's help, there is a real interest in connecting with those same places where the University of Chicago has established campuses around the world to foster exchange between institutions. And so yeah, we're excited to be involved in that. And as I said, I think science really needs to be a worldwide endeavor, and I think especially in marine biology, where there'd be great strength in combining institutional knowledge from multiple marine labs. I think it would be great to get sort of a consortium going. We've been planning on having a meeting that, to bring all the marine labs who are interested in developing new research organisms together. I think we're going to plan on doing that in about a year's time here at the MBL. So look for hopefully an announcement on that.
Great. And I assume that they can reach you through perhaps your website, and certainly our website has information email that people can send inquiries to, and then we would just forward them on to you.
Yeah.
So yeah, people can feel free to reach out to through either website to be in touch with the MBL. A couple other questions relate to, one is microplastics effect on waters. How is that impacting the areas that you work with? And kind of related to that is, radioactivity from Fukushima and nuclear waste. How is that affecting the waters around Japan?
Yeah, I can't really answer the latter question. I don't really have any personal knowledge about that, but I can say a little bit about the microplastics. So in fact, we just had a paper come out from our ecosystems researchers on, the fact that microplastics are really getting into the sediment. They've been getting into the sediment for a long time. And now we're trying to understand the impacts of those microplastics. There's a lot of really good work on, how to get rid of those microplastics and other impacts that they're having. So I think it's very important, and hopefully the other speakers that you'll have from the MBL, they can tell you directly about some of the work that's been going on.
Oh, we're excited to hear more from them. So a couple other questions. One was asking, as a scientist, are we able to do research in two areas? For example, botany and microbiology combined. Is that something that's possible?
Oh, absolutely. So in fact, that's really the direction that science is going, is being interdisciplinary. So in making those kinds of connections. And I think that one that was just asked there, botany and the microbiome, that's one that's fantastically important. So many plants depend on their microbiomes, the bacteria that live with them on their roots, in order to survive, in order to gain nutrients. And so in fact, one of the classic stories is nitrogen fixation by plants, but that's actually done by the microbes that live with the plants, not by the plant itself. But the plant provides the right environment for those microbes to be able to do their job. So there's a absolutely amazing direct interplay between the microbe and the plant. And the only way you can really solve that is by having either collaborations between people that are botanists and microbiome people, or now really training students who actually are very conversant in both fields.
Excellent. And I'm noticing that your colleague Nancy, who's helped us put this program together, put in the Q&A box an email address, director@mbl.edu, for any inquiries from any of the audience that have been coming in. So you have a direct line straight to the MBL. Last question, Nipam, does MBL work with microalgae or macroalgae?
Yeah, there are some projects, and in fact, there's a lot of excitement about using them for food and for biofuel. And so we have researchers that are involved in some of that work.
Dr. Patel, thank you for sharing your expertise and the history of the MBL with our audience. Next week, we'll hold another episode with Dr. Ann Giblin, the Interim Director of the Ecosystem Center at MBL, to discuss the current projections for sea level rise over the next century and what impact high rates of sea level rise could have on coastal ecosystems. We hope to see you next week.